So that's all. That's the material for today: sterilization and disinfection. Talk about the sterilization or disinfection. We want to talk about the two pioneer scientists or doctors who first introduced asepsis condition in clinic. The first one is Ignaz Semmelweis. This is the Hungary. He was a Hungary doctor. That's in 18th century. That's the time. His time was the time before the rising of microbiology, before Pasteur, before Robert Koch. So that's the time before the rising of microbiology. And at that time, people know little about microbiology. People knew little about bacteria. But what he noticed is that if the doctors wash their hands carefully and with some chemical, that can dramatically reduce the rate of infection when moms give birth of kids, give birth of babies. So he noticed that there must be something on the doctor's hands which can cause infection in the mom and cause fever, and finally most of the infected mom will die. So he first introduced the technique of washing hands in clinic, in hospitals. But this concept was raised too early. Most of the people don't accept it, so they don't believe in him, and they thought that this man was mad and insane. So they even sent him to a mental hospital. That's a very sad story. And ironically, before he was sent to hospital, he got injured on one of his fingers, and there was a small wound, and people didn't notice it and didn't treat the wound properly. And several weeks after he was sent to the hospital, he died there. He died of infection, and he did great job on preventing infection, but he died of infection. It's a sad story. So this is the first pioneer doctor, a Hungary doctor. So next guy we are familiar with, his name is Joseph Lister. Joseph Lister. And our first lecture talking about the history of microbiology, we mentioned him. Joseph Lister was a doctor in Britain, a British doctor, a surgeon. He reintroduced the concept of asepsis in surgery, and he tried to sterilize the surgical instruments by boiling them in carbolic acid, and he tried to spray the surgery room with carbolic acid. Another name of carbolic acid is phenol. It's like this molecule. And not long ago, I think even now in some hospitals, we are still using this carbolic acid to spray the floor so it can inhibit the growth of bacteria. So this is Joseph Lister. And from Joseph Lister, this antiseptic technique or asepsis concept grow bigger and bigger. Now in the hospital, we should practice these techniques every day, and this is very important to keep the patients safe and to reduce the chance of infection. So this is Joseph Lister. After Lister, people, with the rising of microbiology, and people notice that a lot of disease are due to infection or due to growth of pathogenic bacteria. And a lot of techniques were developed to either kill or inhibit the growth of bacteria. So that's the techniques about sterilization and disinfection. The purpose of sterilization and disinfection is that first we can destroy pathogens and to prevent their transmission. If there is some pathogen, we can destroy or kill them and prevent their transmission, so that the infection rate will be reduced. The other is that we can reduce or eliminate microorganisms responsible for the contamination of food, water, and other substances. And this is kind of related to our everyday life. And we learn that the microorganisms are everywhere, surrounding us, on us, in us. And some of them can cause disease. And to keep our lifestyle hygiene, we can practice some techniques to keep our drinking water, our food, and other things clean and free, not free from free of bacteria or microorganism, but we can reduce the amount of pathogenic bacteria. For example, the drinking water. You don't want to drink the water from the well directly. You don't want to drink water from the river or lake directly, but we can treat the water with either heat, or we can filter the water to make them clean. So this is the purpose of sterilization and disinfection. First, before we talk about the techniques, we'll first introduce several basic terms, several basic definitions, and we should know what they mean and what's the difference between these definitions. We'll talk about sterilization, disinfection, antisepsis, bacteriostasis, and asepsis. Some of the terms are not covered in detail, but I think you can always check your textbook, the one I referred to you, and you can find the definition of these terms. Okay, the first one is sterilization. Sterilization is a physical or chemical process that completely destroys or removes all forms of microbial life, including pathogenic and non-pathogenic microbes and spores. Here are some points you need to remember. This is a process, so you need to do something. It takes time, the process. You can use either physical or chemical agents, and this process can completely destroy or removes. Completely means all of those microorganisms. Nothing left. You can completely remove or destroy the microbial life. What kind of microbial life? All forms of microbial life. Anything live, anything microbial life, you should get rid of them. This includes either they are pathogenic, which can cause disease, or non-pathogenic. Even they don't cause disease, you kill them or you remove them. And also include spores. This means that you not only kill the vegetative replicating cells, but also kill the endospores, the latent form of life. So this is the complete and most harsh condition to kill anything. So this is sterilization. Another concept is disinfection. Disinfection is a process or act of destroying pathogenic microorganisms, which are capable of giving rise to infection. For example, during this pandemic, and we know that the virus can spread from the surface and can infect some people who touches the surface. So we can use chlorine, and usually we use bleach, or we can also use alcohol to clean contaminated surfaces. For example, the table surface in public area or the doorknobs. We can spray bleach on the doorknobs, or we can spray alcohol or bleach on the glass door in public area, and this will destroy the pathogenic microorganisms, and for example the pathogenic coronavirus, and which can cause infection in human. Okay, so this disinfection is the process to destroy pathogenic microorganisms. Well, the chemical we are using here is called disinfectants, either the bleach or chlorine or alcohol, or some people use isopropanol. Those chemicals, we call them disinfectants. Those products, we call them disinfectants. Okay, these disinfectants cannot necessarily kill spores. It can kill living bacteria. It can kill the replicating bacteria cells, but it cannot kill the endospores. So it's kind of more mild condition. So this is a second term, which is different from the first one, the sterilization, because sterilization is the complete procedure to kill any microorganism, but this disinfection only kills the pathogenic microorganism, not including the spores. Okay, another term we need to keep in mind is the antisepsis. Antisepsis is the method to inhibit or eliminate the growth of microbes on either the skin or other living tissues. For example, before you give a surgery to your patient, you want to clean the skin. You want to treat the skin with some chemical, and this method is to keep the asepsis condition or antisepsis method. And microbes on the skin or living tissues might only be inhibited but not completely killed. They do not necessarily die. On this picture, the doctors, before they give surgery, they need to clean their hands and arms completely. This is also antisepsis. Antiseptic agents should be safe to human and not toxic. And no surprising, it's easy to understand, because you are applying the chemical to the skin. You don't want poison the skin. You don't want destroy the skin. So it should be safe to human skin and not toxic. This is the third concept. Another one is asepsis. Asepsis is the state of being free from microorganisms or preventing contact with microorganisms. Free from or preventing contact with microorganisms. The term asepsis often refers to those practices used to promote or induce asepsis in an operative field in surgery or medicine to prevent infection. So this is like in this picture what you will do. as in this picture. You are a surgeon, you give your patient surgeries. You want to keep the state free from microorganisms. Or during the procedure, you want to be careful. You don't want microorganisms to contaminate the wound. So this is the asepsis. Okay. And to keep the sterilization and disinfection, we can use either physical or chemical procedure, or we can use either physical or chemical agents to keep sterilization and disinfection. First, we'll talk about different physical factors we can use. We'll talk about heat, high temperature. We'll talk about radiation. Radiation is that you can use high energy rays. These high energy rays can destroy molecules in microorganisms, and that can kill the microorganisms. Or we can use like a physical procedure like a filter or filtration. Or you can lower the temperature, because with low temperature the chemical reactions will be reduced, and the procedure of metabolism will be inhibited, and that the growth of bacteria will be inhibited. Or you can also use ultrasonic or sonic vibration to destroy bacteria. And later we'll talk about these procedures one by one in detail. First, we'll learn the method of heat. So you can rise the temperature, so that's the heat. To rise the temperature, you can use different methods. The heat or high temperature can denature microbial enzymes and other proteins. We know that in microorganisms, like in bacteria, it contains a lot of proteins, and some of them are active enzymes. And we know that proteins, to keep them stable, you need a proper temperature. If you increase the temperature high or too high, the protein, the protein is made up of amino acid chains. So the amino acid chains will fold in wrong ways. So it will denature or change from the native state and fold in wrong ways. And this procedure we call denature. After denature or after denaturation, the proteins or enzymes will lose their function. So we can say that the protein is dead. And there are different types of methods to increase the temperature or for heat, like open flame. So you have some fire, like you burn the alcohol or you burn the natural gas. So you have this open flame. You have the fire, which can increase the temperature. Or you can use incineration. Incineration is the procedure of burning. You burn it into ashes. Or you can use a high temperature steam with a pressure. So that's what we used in the lab for autoclaving. You use the autoclave and it can maintain a high pressure so that the temperature can go much higher than 100 degrees. For regular vegetative microorganisms like the replicating bacteria such as E. coli, they can usually be killed at high temperature. Like if you increase the temperature to 50-60 degrees, and they can be killed quickly. So that's not a problem, but the problem is the endospores. We learned the endospores. They have layers of materials to protect them, and those bacterial endospores are very resistant to heat, and so you need to increase the temperature even higher, and you should expose them to high temperature for a longer time to kill them. So So that's for different type of life, you need a different condition to to kill them. The first one we'll talk about is a dry heat. This includes the flaming, incineration, or hot air sterilization. Flaming, that's the open flame, what we mentioned just now. You can like burn alcohol, and you can burn your like metal or rings in the flame, or the test tube in the flame, to keep the opening area sterilized. And incineration, that's a different way. Like in hospitals, if you work there, or in our research lab, we will generate the waste. These waste are not are different from our everyday life waste. They contain medical stuff that we call the medical waste. There might be like blood, there might be microorganisms there, and for those medical waste, we need to treat them with incineration. We need to burn them into ashes. The other one is hot air sterilization. So this procedure, we need to use the oven. So in the oven, you can adjust the temperature to very high, to like 160 degree or over 200 degree. And you put your metal instruments or glasswares, which can be baked. With a high temperature, they are not destroyed. We can put metal instruments or glasswares in the oven, and we bake it for a long time, like 2 hours, 4 hours. This will sterilize this stuff. And with this high temperature, you can also destroy the endotoxin. We learned endotoxin LPS. LPS molecule is pretty stable. So if you want kill LPS, you want destroy the LPS molecule, you need a high temperature, like this in the oven. Okay, another type of heat is moist heat. Moist heat means that it's high temperature, but it also contains moisture. Moisture is the steam of water. It contains water gas, for simple. So the moist heat is usually more effective than the dry heat, because with the help of moist or water molecules, the high temperature can destroy microorganisms with a high efficiency. For example, we can perform the moist heat in different ways. The first one is boiling water. So that's what practiced in China for like maybe thousands of years. And to compare the customer in China and Western countries, you will notice one big difference is that in China, people usually drink the water after boiling. We drink the boiled water. But in Western countries, their hygiene state is better than in China. They will have cold water, which is filtered or treated, so it's pretty clean, so they can drink the cold water. But if you are in some developing countries, and if the hygiene condition is not that good, I would suggest you to boil the water before you drink it, because in the non-boiled water, it might contain a lot of stuff, including bacteria, virus, and even parasites. So you want to make sure that the water you drink is clean, and you should treat it before you drink it. Okay, a second method we call pasteurization, and we introduced this method before, and from the name you can remember, you can recall that this method is created by Louis Pasteur, right? So that is to treat the substances with relatively low temperature, either 60 degree or 70 degree, and with a pretty short time. Like if you treat the milk in at 71 degree, you can only treat the milk for like 15 seconds. So it's pretty short time. And this we call the flash method. This flash heat can kill the microorganism in the milk. Or you can treat the milk with even lower temperature, like 62, 63 degree, and keep the milk at this temperature for 30 minutes. And this is also enough to kill microorganisms in the milk. So this procedure we call pasteurization. Okay, another one we are more familiar with is called autoclaving. Autoclaving, you need to use a machine called or equipment called autoclave. This is one type of autoclave. It's a modern autoclave. You put your stuff in the chamber, and then you close the door and turn the machine on. The machine will increase the temperature and pressure in the chamber. And finally, the temperature can rise to 121 degree. That's 21 degree higher than the boiling water. And you keep the stuff in it for 20 or 30 minutes. And that will kill all forms of life, including the vegetative organisms and endospores. So this is the machine we used a lot in the lab and also in hospitals. In hospitals, you can use autoclave to treat some instruments used for surgery. Okay, another method for moist heat is called fractional sterilization. Fractional sterilization is a long procedure. You can use different techniques. So the general procedure is that if you have something like some liquid, some solution, and the chemicals or molecules in the solution cannot stand the temperature of autoclave. So the temperature cannot be heated over 100 degree. What you can do is that you boil your stuff at the first day. You boil it, and this boiling procedure can increase the temperature to 100 degree, and this will kill all the vegetative cells. But the endospores will survive when you boil it. Then you give your material some time. You incubate them at 37 degree overnight. 37 degree is the proper temperature for endospores to germinate. You incubate them at this proper temperature to allow the endospores to generate into vegetative cells. Then you heat it at 100 degree again to kill this generated vegetative cells. You kill them again, and there might be some endospores left. Then you incubate at 37 degree to allow them to germinate again, and then you heat to kill the germinated bacteria again. And after several turns of this boiling and incubation, you can kill all forms of microorganisms in your substance. So this method we call fractional sterilization. The reason we use this method is because that your stuff cannot stand a high temperature over 100 degree. Okay, so this is moist heat. Okay, so maybe we'll have a short break, and after the break we'll talk about other physical methods for sterilization and disinfection. With the rest of the time, we'll talk about other methods on sterilization and disinfection. So just now we talked about the first physical method of heating. So you increase the temperature. The second method, we can use radiation. Radiation is a physical procedure. You use high energy rays, either UV light or beta ray or X-ray. And these high energy rays can somehow destroy biological molecules. And if the biological molecules is destroyed, or especially DNA molecules are destroyed, the microorganisms will be injured, or sometimes they can be killed. So here are two types of radiation we are talking about. The first one is called ultraviolet radiation, so that's a UV for short. Another one is ionizing radiation. Ionizing radiation is those radiations with higher energy than UV light, such as beta ray, gamma ray. The energy is even higher than UV light, and that rays can break down molecules and to form different types of iron. So that's what we call it ionizing radiation. First, we'll talk about UV radiation. UV radiation, we know that there is UV light in the sunlight, and there is even UV light in the the man-made light we use every day. And you know that our eyes can see the wavelength above 400 nanometer, and those lights lower than 400 nanometer we call UV light. And the most effective wavelength of UV light to influence living cells is from 240 to 280 nanometer. 240 to 180 nanometer, but the optimal wavelength is 260 nanometer. And also another factor we need to consider is the length of exposure. For example, if you expose your skin to UV light for a very short time, probably that's okay. But if you expose it for a long time, it would be a serious problem. And in the lab, usually, especially some old labs, they will install a UV light in the lab, and sometimes they turn on the UV light to kill microorganisms on the surface. But sometimes when people are not careful enough, they didn't notice that the UV light is on. They enter the room and work there for some time, and that can cause damage on your skin or in your eyes, to cause damage in mucosal area. So that's very dangerous. Here I would like to show you a video on UV light and talk about how UV light can cause cancer and aging, before we talk about the effect of UV light on microorganisms. Here's a short video here.
Recently, I made a video about what the world looks like in the ultraviolet. Some things look the same, but what world looks like in the ultraviolet. Some things look the same.
Can you hear the audio?
Hazier, sometimes light colored, but skin looks blotchier, and fake teeth stand out. Whoa. It's not
No? Hold on. How about now?
Do you have a fake tooth, baby?
Yes.
Which one?
But the idea for this video started a long time ago with a question I had, which is, how does non-ionizing radiation, like ultraviolet light, damage skin and lead to cancer?
I do like to tell people that if you want to see how good your skin could look at your butt, because that's an area that doesn't get sun.
The world looks like in the ultraviolet. Some things look the same, but generally it's hazier, sometimes light and dark are flipped. Skin looks blotchier, and fake teeth stand out.
Whoa. Smile for me. Oh my goodness.
Are my teeth black?
Do you have a fake tooth, baby?
Yes.
Which one?
But the idea for this video started a long time ago with a question I had, which is, how does non-ionizing radiation, like ultraviolet light, damage skin and lead to cancer?
I do like to tell people that if you want to see how good your skin could look at your butt, because that's an area that doesn't get sun, and even old people have perfect butts. It's how your skin would age without the sun.
When I thought about what UV light must be doing to our cells and our DNA, I imagined it would be getting in there and breaking all these bonds, and that would be the problem. But in fact, it's kind of the opposite. UV actually facilitates the formation of covalent bonds in our DNA that shouldn't be there.
It comes in and it kind of creates these unauthorized bonds between the bases called pyrimidine dimers. Instead of having them bonded across, it bonds the one next to each other, if that makes sense.
Oh.
Yeah.
So what happens is two thymines, which are side by side, can actually get covalently bonded together. And that creates a lesion on the DNA. You can think of it like a little bump that makes the DNA hard to read and work with.
That's not enough in itself to cause skin cancer. If that were the case, we would just be covered in skin cancers. Then the next step is that we have to repair it. And in the repair, that's when you form mutations that could eventually lead to cancer through a bunch of different pathways. And there's actually people who have specific genetic issues that they can't repair these issues, and they are covered in skin cancer.
UV has other effects on the body, like it suppresses the immune system. So as Haley says, it's a double whammy.
There's also kind of a double whammy because UV not only induces these unauthorized bonds, it actually tamps down our natural immune system. So one of the best ways we fight cancer is through our immune system. We have constant immune surveillance through our skin. So not only is sunlight creating these problems, it's actually
reducing our ability
to fix them. So unfair.
UV light also causes us to visibly age. It's called photoaging. I should clear up a misconception here that I had, which was I thought glass blocks all ultraviolet light. So I thought if you're inside a car, for example, you really don't need to put on sunscreen because you're not being exposed to UV light.
So the car does not block UVA, it's glass. It does block most UVB, but you're still susceptible to those sunspots and those wrinkles through the glass. And a lot of my patients, if you look and you compare their hands, the left is more wrinkled or has more sunspots on it because they're driving.
So they're sticking that hand
out there. Truckers, absolutely, you can see it the worst. And there's a really famous picture of someone whose left side of their face is significantly more, as we would call it, like photo age or old looking than the other side of their face. So that's mostly UVA. And UVA, as I said, it's a longer wavelength, penetrates a little bit deeper. So wrinkles mostly come from destruction of our collagen and elastin, which is natural fibers that hold up the scaffolding of our skin. So with age, the cells that secrete collagen, they're called fibroblasts, they stop producing it as much. And what the sun does is it breaks down the existing collagen and elastin we have. So fibroblasts really like to be stretched. They like to be pulled. And collagen does that collagen matrix. So the sun damages the collagen fibers, leading to less stretch on fibroblasts and less production of collagen, which means that we look a little more saggy. I know, it's very depressing. So sun protection helps the degradation of those collagen and elastin fibers.
You know, there are a lot of creams on the market that claim to be anti-aging, but the one that really works is sunscreen. Preventing ultraviolet light from hitting your skin is the best thing you can do to avoid skin cancer and to avoid photoaging. So yeah, it's good advice. Use sunscreen. Hey, I'm on set for Jurassic World Fallen Kingdom, and I'm here filming Sci-Fi Test Lab, where we try to recreate some dinosaur parts out of machines.
Okay, so that's the effect of UV light on cancer and aging, and that's on our human. And this gives us some idea that when people when you go out or even stay at home, try to protect yourself from UV light, because UVA can go through the glass window, and after a long time exposure, it can cause aging on your skin. So this is especially a good suggest for girls in the class. Okay, after talking about the effect of UV light on human, and we are focusing on the effect of UV light on microorganisms, what they cause in bacteria. So that's what we learn here. It's also mentioned in the video that when the UV light shines on the DNA molecule, it causes the DNA molecule to form a TT dimer. So it's a thymidine-thymidine dimer. It's a thymine-thymine dimer. And you know, when your DNA has this structure, this is a covalent a chemical bond, your cell or the bacterial cell will try to fix it, try to fix the dimer. But if there are too many dimers in the DNA, the bacteria will die. So that's the effect of UV light on bacteria. We can use UV light to sterilize the air, or we can use it to sterilize the surface of objects. For example, in the tissue culture room, you turn on the UV light. The UV light can kill any bacteria floating in the air, or that it can kill the bacteria on the surface of the table. So that's useful. But the disadvantage of UV light is that the penetration ability is poor, very poor. It can only shine on the surface. It cannot go deep inside the objects. So that's because the UV energy is still not high enough. It can damage our eyes, it can cause burns, and cause mutation of our skin cells. So that's the dangerous side of UV light. So you don't want to expose yourself under UV light for a long time. When you go outside for to play in the playground, or if you go to a tour, make sure that you bring your sunscreen with you and apply it frequently and to protect you. So that's the UV light and the effects of UV light on both our human and also on microorganisms. Here I will show you a video. This video is an experiment. They did this experiment to test the effect of UV light on bacteria, and you can see how effectively the UV light can kill bacterial cells. First, you spread the bacteria, or you streak the bacteria on the surface of a culture dish. You know, this is a solid agar, solid medium. And then you cover half of the plate with a piece of paper. And then you shine the plate under UV light for different times. This time they expose the bacteria to UV light for 15 seconds. Then the dish is cultured at 37 degrees to allow the bacteria to grow. And you can see the bacteria can grow all over the plate. The second plate, you spread the bacteria, and after 30 seconds of exposure, you can see that only half of the plate can grow bacteria. The other half, you have only a few colonies survived. So you killed most of the bacteria. And this time, 1 minute exposure. And you grow the plate, and only 2 colonies survived. So this is 1 minute. And if you expose to UV light for 2 minutes, and this time no bacteria survived. So 2 minutes UV light exposure can kill all the bacteria on the left side. And also the other thing you should keep in mind that they only used a piece of paper to block the UV light, which means that the UV light cannot penetrate through the paper. So that's a very thin layer of paper. So UV light penetration ability of UV light is very poor. Okay, so that's the effect of UV light on bacteria. So, but after we talk about the UV light radiation, we'll talk a little bit about the ionizing radiation. This is a radiation with either X-ray, gamma ray, or beta ray, and these are the high energy radiation. They have much higher energy and can penetrate deeper than the UV light. We can use gamma ray to sterilize pharmaceutical or disposable medical supplies, such as syringes, surgical gloves, catheters, or sutures. So those are the material you will use during a surgery. And for some of them, you cannot autoclave them, but you can use this ionizing radiation to sterilize them. Okay. Another physical method we can use is filtration. Filtration, or that another word is we can call it filter. It's a very simple procedure that you allow some liquid or air to go through a filter. Here is the filter installed. To go through a filter. On the filter, there are very tiny pores. The pore size is usually around 2.22 micrometer. And we know that this size is much smaller than bacteria, so that bacteria cannot pass through the filter. So the flow through will free of bacteria. Anything past the filter will be bacteria free. So this is a procedure we call filtration. For filtration, you can set up the equipment in different ways. This is the bigger way, flask. You can use filter and filter like a bigger volume. You can also use this setup on a syringe. And this is a setup of the filter. This is the filter. You install it in the middle and turn it tight. And you can use this syringe to push and filter the solution in different ways for filtration. Okay, so this is the filter after filtration. After you filter some solution, you take the filter out and then you observe it under a scanning electron microscope. And you can see that there are some round dots, little balls on the filter. These are the bacterial cells. So the bacterial cells cannot pass through the filter but stick to the surface. So these are the bacterial cells. And these little dots, these black dots, are actually the pores of the filter. So these are the pores on the filter. So it's so small it can block the bacterial cells to go through. So this is a way for filtration. Another way is freezing. You know, we learned that if you culture the bacteria, you need to provide a proper temperature, and at some optimal temperature, the bacteria can grow well. And if the temperature is too low or too high, they won't grow well. And freezing is the extreme end of the low temperature. You lower the temperature to like -20, -30, or even -80 degree. At that temperature, it will be frozen. You can slow down the microbial metabolism, and slow down the metabolism, you can inhibit the microbial growth. So this is a way that we can keep the bacteria or inhibit the bacteria from growing. Okay. And this is a way that we can keep our sample fresh, or either for research or for clinic practice. If you have something, if you don't want bacteria to grow on the sample, you can freeze it. You can put it into the freezer and keep it for a while. So this is freezing, also a physical method. Okay. Then after we talk about all the physical methods, we'll talk about the chemical method for sterilization and disinfection. These chemicals or chemical agents are antimicrobial agents. They have the antimicrobial activities to like kill or inhibit the growth of bacteria. They can damage the lipids bilayer on the membrane, or that they can damage proteins on the membrane, resulting in the leakage of the cellular material. If you make pores on the cytoplasm membrane, and water can get into the cell, and the contents of the cell will come out. So the microorganism will be lysed, they will die. So this is one way that the chemicals work on the membrane. The other one is that they can denature the microbial enzymes and other proteins by disrupting the hydrogen bonds or the disulfide bonds. And if you learn biochemistry, you know that for the protein to function properly, the amino acids chain will fold correctly. And after folding, there might be hydrogen bonds or disulfide bonds in the protein molecule to keep the molecule in the native form so that the protein can function. But some chemicals, they can denature these protein molecules. They can disrupting these bonds so that the protein will be killed, will be denatured. So this is another way. there are different types of chemicals we can use for sterilization and disinfection. The first one is alcohol. This includes different types of alcohols like ethyl alcohol, so that's alcohol in wine, in liquor, and also isopropanol. Isopropanol is alcohol with 3 carbon element, and both of them can kill or can inhibit bacteria, but they have no effect on endospores and non-enveloped viruses. They can kill the replicating bacterial cells, but they cannot kill endospores. So this is first one, alcohol. The second one is phenol. This is the one we mentioned just now. Another name of phenol is carbolic acid. It can alter membrane permeability and denature proteins on the membrane. So it can inhibit bacterial growth or kill bacteria, but it has no effect on endospores. And like alcohol, it cannot kill endospores. This is phenol. Okay. A third one is heavy metal derivatives. Heavy metal derivatives. And during our humans' history, our ancestors have been using heavy metal to keep either the wine or the food fresh for a long time. The most popular one is silver. Silver is not that expensive, and it's more very soft. You can change the shape, so you can use silver to make pans, to make kettles, and you can keep wines in it, and that can keep it fresh for a longer time. And still, we don't know exactly the mechanism, how it works, but it can keep it fresh for a longer time. So that's the heavy metal. But again, heavy metal cannot affect on endospores. Okay, another group of molecules we call peroxide. This includes hydrogen peroxide or chlorine. This peroxide is more useful, more effective. They can be effective on endospores. They can kill the non-replicating endospores. Okay, the last step is aldehyde, including glutaraldehyde or formaldehyde. So these chemicals can kill vegetative bacteria and also endospores and viruses. And usually, in our research lab, we use these chemicals as a fixing agent to fix different types of cells. Okay, so these are the different types of chemicals we can use to to perform sterilization and disinfection. So basically, that's what we'll cover today. That's what we are talking about on this topic. Before the end of the lecture, I would like to show you some short videos, and those videos may help you to understand our material better, and it will give you a better impression and help you go through our lecture and our textbook. Maybe we can play the first one.
Sanitizers versus disinfectants. Do you know the difference? The desks in your office, the lunchroom tables, the bathroom. What product is safe to get rid of the germs? To sanitize means to make clean and hygienic. You are reducing microorganisms to a safe level. Sanitizers work quickly, reducing contamination by 99.9% in 30 seconds. The food industry most often uses sanitizing procedures. After using sanitizers, surfaces are safe for food contact after the surface has air dried. Thank you, Jamie, for demonstrating sanitize. Disinfectants are different. Disinfectants destroy infectious fungi and bacteria on hard surfaces. This takes some time. Treated areas must stay wet for 10 minutes, and disinfectants do not recognize the difference between good and bad bacteria. It destroys it all. That's a problem for us. There are millions of good bacteria swimming inside of us all that we need to stay healthy. Disinfectants are best for general public areas, locker rooms, and restrooms. Thanks, Sherry. NABC works great. Here are some other great disinfectants we carry. If you do use a disinfectant in a food contact area, the treated surface must be rinsed well before reuse. As with any chemical, always follow the manufacturer's directions on the bottle and have the SDS handy in case of emergencies. Click on the products for more information or call us today.
Okay, so that's the first. We didn't cover the sanitizing. That's for like everyday life, not in our microbiology class or clinical practice. And then we can watch this, the sterilization techniques.
Alright, I'd like to talk about sterilization techniques. Now, sterilization may seem like a trivial aspect of microbiology, but it's extremely important in the laboratory. And the first time I ever made media in the laboratory, I ended up basically contaminating all of it when I was done, because I just didn't really understand the sterilization techniques and how to work under sterile conditions. So it's extremely important to understand what goes on there and why we do it. Obviously, if you're trying to grow a particular bacteria on a media, on some kind of media, you don't want other bacteria growing on it. You don't want other viruses around it. You don't want anything else. You don't want fungi. You don't want, whatever, protozoa. You just don't want these things to grow because you want to eventually obtain most likely a pure sample. So with the sterilization techniques, the one probably most of us are most familiar with is pasteurization, and that's common if you drink juice or milk, maybe is the best example, where basically it's a process of heating to 63 degrees Celsius for 30 minutes, okay? And it doesn't kill all the cells. But what it does do is it kills things like tuberculosis and some of the other heat-resistant but non-spore-forming bacteria, okay? So it's heating it long enough to kill heat-resistant bacteria, but ones that don't form spores, because dealing with endospores is really difficult, and the only way to deal with spore formers and to ensure that you've killed all spores is to use one of these. And this is an autoclave, okay? And the other thing about pasteurization is it leaves food tasting normal. We know this because we drink milk or whatever, and it tastes normal. It doesn't taste weird in any way. So, but it doesn't kill all cells. Okay, so some things are still left alive. Now the autoclave is basically a way of completely sterilizing everything, okay? It kills all bacteria, okay? It kills all bacteria, kills all endospores, and that's what you do when you're making media, or if you're cleaning some pipette tips or something like that, you're going to use the autoclave. And essentially what the autoclave is, everyone's familiar with this, it's just a steam cooker, okay? These are common in kitchens. So it's just a steam cooker. You're heating to 121 degrees Celsius, at 15 psi for 20 minutes, okay? It kills all bacteria, it kills all endospores. So an extremely important piece in any laboratory. Another way to deal with killing bacteria or sterilizing instruments or anything like that is to use radiation, okay? So basically, it damages the DNA, it causes mutations, and depending on where these mutations occur, those mutations can be lethal or maybe they're not lethal. But in most cases, they cause some kind of damage to the organism that, that decreases its ability to continue surviving. So radiation is another effective way of sterilizing things. So you can use UV light, you can use X-rays or gamma rays being the, obviously the strongest. But in a lot of cases, it's UV light. So a few more sterilization techniques. Now this is probably one of the more interesting and useful ones I actually have pictured here, and you might not know what it is, but essentially it's a filter, okay? And what happens here is infiltration. It's used in cases where heat will destroy the organism being, used. So if you're working with a heat-sensitive bacteria, you're not going to want to use an autoclave. You're not going to want to use other kinds of heat or radiation. You might want to just use something where you can simply filter it through. Okay, and essentially whatever you grow your bacteria up, and you pass it right through this filter. Okay, you pass it right through, probably in a liquid media, right through this filter, and basically the pore size is 0.2 micrometers. And that 0.2 micrometers is good enough to catch any bacteria. Okay, so no bacteria are going to pass through the filter. But at the same time, other things will, okay? Other things will. So things that might pass through the filter will be things like viruses, okay? So viruses can pass through because they're much smaller. And it's unlikely, though, that you're going to find, have viruses laying around on your bench or something like that at the lab. I mean, maybe you will, maybe you won't. But in most cases, it's something that's kind of insignificant, so you don't really need to consider it or worry about it. Also, another common thing, everybody has these in their lab, and they have refrigerators and freezers of various kinds. So you might have a refrigerator if you're putting something in that you just want to, say, not have any chemistry occurring in, but you just want to keep it from heating up or gaining, or whatever the case is, or you want to slow the growth. And that's what cold temperatures can be used for. And of course, more long-term storage, you might use like a negative 80 degrees Celsius freezer. So it slows bacterial growth. It does not kill the bacteria. That's the important point. Cold temperatures are not going to kill it unless they're really, really cold. I mean, they'd have to be really cold. But the point being is they don't kill bacteria under normal conditions. And in talking about this, I guess I've been a little bit vague. There's different forms going on here. Not everything is so-called sterilization. So sterilization is the process by which all living cells, spores, viruses, etc., are destroyed on an object. Okay, so if you have an object, let's say you have a spatula from lab, you want to disinfect it, you want to remove all spores, everything from it. You're going to put that in the autoclave. It's going to kill all the spores, all the viruses, all the cells. Everything dies. That's sterilization. Okay. Now, disinfection is a little different, okay? So disinfection is the killing or removal of disease-producing organisms from inanimate surfaces, okay? So you want to remove the disease-forming organisms from inanimate surfaces. Think of a hospital. Think of a surgical tool, a scalpel, for instance. I don't know. Might be a great example. You want to disinfect it. You want to get any kind of disease-producing organisms off of it, okay? That might be something. A place where you might see this disinfection occurring. So you want all the disease-causing organisms off of the surface. It does not necessarily result in sterilization, though, okay? So the pathogens are killed, but other microbes may survive. Now, antisepsis is similar to disinfection, okay? But it applies to removing pathogens from the surface of living tissue, okay? Obviously, you're not going to want to use an antiseptic that's really, really strong, because it's got to go on human tissue, okay? It could damage human tissue. It could damage the tissue of the organism. You don't want to do that, obviously. So the disinfectant agents that you might use on, say, your laboratory bench are not going to be the same that you're going to use on the human body, okay? They're just simply not going to be as harsh, okay? And so they're usually not as toxic as the disinfectants, and that would frequently damage living tissue. So damage to living tissue can definitely occur if you're using a disinfectant when you should be using an antiseptic. And another interesting thing here is that antibacterial agents can be one or the other of bacteriostatic or bactericidal. Okay, basically what bacteriostatic means is that they're going to inhibit the growth of the bacteria, but not necessarily kill them. So they inhibit growth, don't kill them. But bactericidal chemical substances that kill microbes. Okay, so it depends on what you're working with.
Okay, that's all for our lecture. We do have more videos for you. You can always log into our class on Superstar, and there you can download our PPT file and you can watch the videos online.